Hello. Let's see.
Hi, Lauren. How are you today?
Good, how are you?
I'm doing good. I'm doing okay today.
Well, do you have any questions for me? So you moved to Monday, Wednesday, right?
Yes. So I'm in Monday, Wednesday. My case study, it was a bit of a shock for me. I don't think it was a shock, but I think it's just, I don't know. I think I am realizing it's like everything's hitting me. I don't know how to make it make sense, but it's like this is the major that you picked. And this is what you go to school for now. So I think that the level of professionalism during my case study performance wasn't there, and I honestly was thinking it was honestly because of my understanding of peptoglycan, the peptoglycan layer, and understanding okay, so why is your gram stain gram positive? And so yeah.
Did you want me to
So
go
I think
over
just
that?
the layer.
Okay, yeah. Okay. So let me go ahead and draw the layer for you. I have a nifty little whiteboard. So I'm going to draw a gram positive and a gram negative, but I'm going to tell you what each one is. You have to tell me which one's which. And you can probably see the board fine, right? It's a lot smaller on the screen.
Okay.
So that's just my divider. So these are my teichoic acids. That's my peptidoglycan. And then I have my there we go. Now for the other part. Kind of got lazy towards the end. So this one, this big block is my peptidoglycan. These are my teichoic acid, and this is a phospholipid bilayer. And then I have another phospholipid bilayer here, a thin peptidoglycan. This is going to be my periplasmic space. And then I have another phospholipid bilayer here. Now, which one's gram positive and which one's gram negative?
The one with the thick peptidoglycan is gram positive?
Yes, right here. Gram positive, and so this one's got to be gram negative, right? So they also so how the gram stain works is going to be influenced by their physical characteristics, right? So when we have, I hope that my purple shows up, but when we have our steps of our when we have the steps of our gram stain, what happens, right? First, we have our crystal violet, right? So if we have our crystal violet and it's going in, it's going to go past these layers. So we're going to have crystal violet here, right? It's going to go past the peptidoglycan, okay? So when our crystal violet goes into each of these, they're going to look purple, right? If we just do crystal violet, they're gonna look purple. Okay. So then, if we go in with our iodine mordant, so this one's gonna be iodine. Iodine. There we go. If we go with our iodine mordant, it's going to cause a complex, right? Gonna kind of attach. It's going to solidify this here. It's gonna kind of join this purple. It's gonna look a little less brilliant of a purple, but it's still gonna look purple. Okay. And then if we go in with our next step, which is our depolarizer, and that's going to be our ethanol. If we go in with our ethanol, now that does something special here. It's going to basically, because of how thin this peptidoglycan layer is, it's going to take all of this out. So it's not going to be there anymore. But this, because of how thick the peptidoglycan layer is, and because the mordant applied past this layer, we're not going to see the color lift in our gram positive. So our ethanol lifted the color here. So at this stage, at this first purple line, everything was purple. Here, everything's still purple. We add the depolarizer, gram positive is going to be purple, but gram negative is going to now look colorless. So now we'll go in with our last step, and that's going to be our saffron. I'm probably writing almost off, but our saffron is going to come in. It's going to just probably make this a little bit more purple. It's not really going to change the color up a lot, but it is going to apply a counter stain here. So now this is going to appear pink, and this is going to stay purple. And because of their physical differences, they stain differently. Does that make sense?
That does make sense. That is really helpful. It makes a lot more sense. But I think you're talking about like the thin layer on the gram negative. And what is that called again?
This thin layer right here.
Mm-hmm.
So that thin layer is the peptidoglycan that is also the difference here. These two are the same.
Okay.
Except in gram positive, it's very thick.
Okay.
In gram negative, it's really thin. Also, this may or may not be on the exam, but for our gram negative, she's going to talk about how they're a little bit more resistant to antibiotics. And the reason is that an antibiotic, I'm just going to kind of do it like this. If we have an antibiotic and it comes in to this layer, it may or may not come in the first place, because remember, these are phospholipid bilayers. They're selective, right? They have selective permeability. So if they're going to come in here, they might stay here for a second, but they're going to get spat right back out most of the time, because the bacteria realizes that's not something it wants. When an antibiotic here hits this, sometimes it targets peptidoglycan synthesis. So if it's going to touch the peptidoglycan right off the bat, it might damage this whole thing, right? All of a sudden, this is damaged, this is exposed. So this makes it more vulnerable. This is the only layer it has now. With this, the antibiotic can't even get by the first layer, let alone the second or third. So we have we're gonna have in both these phospholipid bilayers, but the difference is this one has two. It has an outer membrane and then the inner. So gram positive has thick peptidoglycan, it has teichoic acids and no periplasmic space. And then over here, and also it doesn't have an outer membrane like this one. So over here, this one does have an outer membrane, a thin peptidoglycan, a periplasmic space, and then the normal phospholipid inner bilayer. And this one also has no teichoic acids. because the teichoic acids sit right here in the peptidoglycan. This one, it doesn't have anything. It's just peptidoglycan. Is this okay? Does this
Yeah,
make
that's
sense?
good.
Okay.
Yes. Thank
One
you.
other thing, is it okay if I erase this?
Mm-hmm.
One other thing that Dr. B, I was asking her about, because I remember specifically my year that I didn't get too many questions about the peptidoglycan itself. I didn't get a whole lot of questions on my exam, but she said that I just didn't get the version with a whole lot of questions about it, which means that it is going to be a staple in your guys' questions. So we wanted to take a look at, yes, I can. I can go over that electron transport chain. So one other thing that I did want to touch on was the NAG-NAM in the peptidoglycan. So the main things you need to know is that we're going to have an alternating sequence of NAG-NAM. I can write faster. We're going to have this alternating sequence. We're going to have, she doesn't need you to know the name specifically, but this is going to be a, I think it's a tetra something. I'll get the actual name, but it's going to be 4 proteins. Tetraprotein? I don't know. Let me look it up for you guys so I'm telling you the right thing. But it's going to have 4 things right here, right? And that's going to be attached to one side. And then we're going to have 4 right here. And they're going to have this peptide bond cross-loop. Let me go ahead and pull up the there we go. It's loading right now, but these are going to be the same. You don't really need to know exactly what they are, but know that they are alternating. It's going to have the 4 right here and then a peptide cross link. I'll kind of show you what it is. Let's see. Here it is. Tetrapeptide, they're peptides. They're the building blocks of proteins, but so yes. Let me share my screen real quick. Also know that the NAG-NAM are very similar to glucose, right? So this is kind of what I was drawing out here. You're gonna have alternating NAG-NAM tetrapeptide with a peptide cross link. And she also wants you guys to understand that they draw similarities in their structure. So this is just, so the NAG-NAMs are just going to be kind of derivatives of glucose. You see how they're very similar. Okay. Yep. So I think that's all she wanted you guys to know on that one. Let me pull up the PowerPoint slide for the electron transport chain. That one, I'm gonna be honest, kind of threw me for a loop too. Let's see. There we go. Let me change my share. There we go. Okay. Let me erase this real quick. But does this make sense? The alternating NAG-NAM. This seems familiar. Does that seem good? I'm gonna go with the yes. It's on the slides. I just tried to simplify it for you guys. Because sometimes those big sciencey words, they intimidate me. And you don't need to know some of them. Okay. It's kind of some of this information is you just kind of have to take it at face value. You can't really be like, well, why is it NAGNAM? Why is it like that? Just know it's NAGNAM, it's alternating. They have the 4 tetrapeptides and they're connected by a cross peptide link. I think that's all you really need to know. And then that NAGNAM are derivatives of glucose. They have very similar chemical structures. Okay, so let's take a look at the electron transport chain. So this is the last step in respiration, right? Let me pull up that. Here it is. So, right, we have either if it's fermentation or respiration, they're going to go through glycolysis, right? And so when they go to glycolysis, they're going to have pyruvic acid. Pyruvic acid is going to go to the acetyl coenzyme A cycle, and then we're going to go through the Krebs cycle, and that in and of itself is going to produce some ATP, right? Some energy. Then with our NADHs, they're going to join together in forces and go along the electron transport chain. Okay, so that's kind of our lineage here. You will have to know this a little bit more in depth. It's not so much face value as what is its metabolism. And remember that respiration is always aerobic. So it's kind of like this. This is kind of what you need to know in a very good summary. And then know this and this. This synthesis of acetyl coenzyme A and the Krebs cycle, know that in a little bit more depth. You'll probably need to know what goes in and what goes out for every single step. And that's just memorization. Let's see. So we got the Krebs cycle and then all the way down here, electron transport chain. So this is where most of the ATP comes from, right? So the best way I can kind of show you is this is our cell wall, right? This is the innermost layer to the bacteria, right? Or for the cells in general. This is the innermost layer. What kind of happens is, I'll draw it in a different color, is that all of a sudden we have, there you go, electrons kind of doing this dance. They go in and out, in and out, in and out, right? And then this is what the chain that we build, okay? So this is the basic kind of pathway that they take. So, and these are all redox reactions. So we have a redox reaction, redox reaction, redox reaction. Let's see. So yes, if we look at the electron transport chain in prokaryotes, so bacteria, for example, if we look at it, it's going to look like this. But if we look at it in a eukaryotic organism, we're going to have the powerhouse of the cell, and then we have these little lines, right? These are all called cristae. I hope I spelled that right. All these cristae have their own ribosomes, their own protein factors. Okay. So the electron transport chain happens here in eukaryotes. Prokaryotes is the cytoplasmic membrane. For the sake of this class, you'll need to know the process in prokaryotes better than you do eukaryotes, right? And this is very similar to the process that you guys saw in anatomy and physiology. You guys will see that kind of happen again here. So this is kind of what I was trying to simplify here. So this is going to be a good a good kind of summary as to what's happening. So we have NADH and FADH2 bringing electrons and protons in. So these guys are coming in.
You don't have that like all it's doing is specifically.
Okay, so these guys are coming in to our cytoplasmic membrane. Let's see. This is from okay. So if these guys are coming in, where are they going? Let's see. I don't want to tell you, I don't want to word this wrong. I want to word it how she worded it in her lecture. So
Okay, thank you so much.
okay. So among this cytoplasmic membrane are going to be these carrier proteins. So these are all.
Oh, no. I'm sorry, because I'm 143. You're 141.
So these are the carrier proteins. They're traveling. This is kind of misleading, so I apologize. But it is kind of the pathway they do take inside of the membrane. What's really going to happen is if these guys go in and they're going to jump.
They're actually set on. Kind of, it's kind of like
I don't want to confuse you guys. Okay, so they're gonna jump, and they're gonna keep going. And I would relate this to, like a snowball going down a hill. It's just gonna keep going, and the longer it goes, the more energy it gets. Okay. All you need to know for what I've said so far are what are some carrier proteins, and I have that right here. So we have flavoproteins, ubiquinolones, metal-containing proteins, and cytochromes. Okay. Let me see if I can. I'm gonna try to enlarge this picture too, because if you just redraw this, you get a pretty good understanding of how it works. Oh, here's a better here. Speak of the devil, right? So we have our NADH and FADH coming in, right? And like I said, these carrier proteins like ubiquinolone right here, they're chilling in here. They come in and they lose an electron. So these guys, these are their like heightened state, right? If they're NADH and FADH2, they're kind of in a positive state. Or they're in like a neutral state, but once they leave, they're in their normal like negative state. So when they come in here, they're going to jump out here, and NADH is going to now be NAD, and FADH is going to be just FAD. Let me double check. FAD, yeah, FAD. And it's going to be negative. So these guys just kind of hold energy and give it to to the carrier proteins, and then they break them up a little bit and spit out hydrogen. And that's where our energy containing molecules are. So by breaking those bonds, it releases energy. We broke a bond in here. Now we are going to have energy. We're going to have, I'm actually not going to do pluses because it's an electron. We're going to have energy, right? Energy, broke a bond here, energy, broke a bond here, energy. These are all electrons. We get electrons from breaking these guys up. Does that make sense? So these are products from the Krebs cycle going into the cytoplasmic membrane. When they get into the cytoplasmic membrane, they're going to meet these carrier proteins. Carrier proteins? Yes, carrier proteins. They're going to meet these carrier proteins and they're going to lose some of their hydrogen. And by the breaking of those bonds, they gain electrons. And as they keep going, they're gaining more and more electrons. Okay, until they finally meet the molecule that they're going to bond with. So in the electron transport chain, the final electron receiver, let's see, that was kind of what I was explaining. It was over here somewhere. Let me find it. Here it is. Here it is. So the final electron acceptor for aerobic respiration is going to be oxygen. Okay, and then if we see any other electron transport chains happening, these are the final electron acceptors. But for the sake of aerobic respiration and the electron transport chain, which is what happens 99% of the time, it's going to be oxygen. And we see that here, right? It forms a H2O complex, right? You kind of see what's happening. And this creates energy. Let's see. Energy from each electron being passed down the chain is used to pump protons from one side of the membrane to the other. So all of a sudden, yep, oh yes, this is also what makes the inside of our cells negative compared to the outside, which is positive, right? So if we have a positive charge getting pumped out here, all of a sudden we got a lot. Because I mean, these cells are always producing metabolism or going through a metabolic process. If we weren't going through metabolic processes, then, you know, we would stop existing eventually. So they're always making this positive and always keeping this negative, right? NAD negative, FAD negative. Got the lack of electrons on this side, lack of hydrogen on this side. Because when these are broken, the NAD and the FAD, they can be reused. They can go back into the Krebs cycle. But the hydrogen just gets pumped out. And the hydrogen is what makes all these positives. Okay. So NADH, FADH2 comes in. They break and give us electrons. They pump out the hydrogen. The electrons keep going and going and going. And then all across these carrier proteins, and then eventually they join up with oxygen at the end for aerobic respiration. I know that's a lot of information. I recommend you guys downloading the PowerPoints and looking at her notes at the bottom, because it kind of just lays out the steps. And it gives you a lot of good visuals you can look back to. Does that make sense? I try to draw it out as best I can. I know, Anna, that you
feel
asked this
like that-
initially. Go on, I'm sorry.
No, I feel like it helps that you showed us this part of the slide so that like we can really focus on this and like draw it out in the future. And yeah, her notes are helpful.
Good.
Yeah.
Yeah, no problem. And then Anna, I'm could I have you, if you want to direct message me, just your student ID number so I can sign you in. It's more for me. It's not a track record for you, it's just for me to make sure I actually have people coming. But yeah, so one other study tip, because I know that, you know, this weekend is basically cram weekend. What I would suggest you guys do is kind of go through these slides And if you kind of understand what's happening, if you don't see anything that's like too crazy, I would recommend just going through that to study for the lecture portion. And then to answer your question, this is the end of aerobic respiration. I got the question, does this prepare you for aerobic respiration? So let's take another look at the, I'm going to always reference this because this is one of the better summary slides. So this is the whole cycle of respiration is glycolysis to the acetyl coenzyme A, Krebs cycle, electron transport chain. These all work together to produce respiration, right? And then fermentation is the other part of that. That's aerobic, right? So these both undergo glycolysis, except this one just ferments as the end products. Respiration is a lot more complicated. And these are both catabolism. Let's see. I hope that answered your question. But one last thing before you guys go, I would like to just show you guys one other study tip that ignore that, sorry. One other study tip that I would recommend if you guys feel overwhelmed or stressed out for the lab portion. I will give you kind of the end all be all, the magnum opus. Oh, that is the wrong thing. My bad. If you study this, you're gonna be fine on the lab portion of the exam. If it'll load. And then you can devote a little bit more time to the lecture portion because there's going to be, I mean, you already dedicate like, what is it, 8 hours a week? No, 6 hours a week. You guys dedicate 6 hours a week already to lab material, right? And maybe there's a little bit of, you know, lecture stuff sprinkled in, but it is going to be mostly lab material that you're going over. But you only spend 3 hours on the lecture material. And arguably, there's a little bit more lecture material than lab material most of the time. So this is what I would recommend you guys use in order to review the lecture. Because this is kind of just, it's all in one place. With lecture, I feel like you have to kind of pick through it with a fine-tooth comb to figure out what you need to do. So a lot of people didn't utilize this, and it's an amazing resource that is just here for you guys. So this is your study guide for the practical exam. Look, it says things like, what was the purpose of streaking for the isolation? Answer that question and you're good on that. You know, there's going to be a lot of like, how is scanning and tunneling microscopy different, and what structures is it useful for in viewing? If you can answer these questions, no problem, you're going to have a relatively good time on that lab portion. You know, it tells you the skills that you physically need to know. And then it also says define, be able to, define selective media, define differential media. It asks you specific questions based on what plates you guys have been working with, okay? Use this as your consultation, right? Use this and then answer the questions and define what you need to define, write it out or type it out, but use it. And then also, this offers a little bit more on the imaging side of things, of what you might need to visually be able to recognize and kind of test you in some different ways. I know that if you look back on the slides, you may be able to draw a connection between, oh, well, she said that this was this, you know. But some of these pictures are things that she didn't show you. She showed you this, I know that. There's not a whole lot of different ways to draw this, but there are a few ways to show you different types of microscopes, right? So if you can say this is this, this is this, and this is this, and that's what the microscopes come from, or where those images come from, then you're set. So take a look at these. If you can see best something, if you can tell me what technique this is, if you can tell me what metabolic processes or the oxygen requirements needed for each of these are, you're set. But use this in accordance with the study guide, and you'll be good. That's all that you really need to that's all you really need to be able to do for the lab section. She's not going to really pull anything too far out of left field. You're going to at least have talked about it once or twice. But those are my study tips. And this weekend is really, just cram weekend, I think. And then you come on Monday or Tuesday with questions. And then I believe you guys take your exam on Wednesday/Thursday, or is it Monday and Tuesday?
I don't personally know, but I don't know if the other person in here knows, but I was gonna ask you that, like, what day is our exam?
Let me see. Because I have your guys' syllabus, which is also a great, here we go,
I think maybe Wednesday.
Wednesday. If you guys are ever questioning, oh no, I opened the syllabus, not the schedule. If you guys are ever questioning, like when something is, or like how much time do I have to prepare, when is something due, she follows the schedule pretty religiously, I will tell you that. Ooh, I jumped all the way down. So exam 1 will be, let's see, I have something in the way. Wednesday and Thursday. So study all weekend, bring your questions Tuesday and Monday, and just go for it. And another little piece of advice that I have is that once you walk into the classroom the day of the exam, don't try to scramble for information. Don't try to be like, oh, well, do I know this? It's either you know it or you don't at a certain point, right? Don't stress yourself up to the last minute and then just stay stressed out through the whole exam. I would rather you guys study and decompress, and then maybe quiz yourself here and there on the walk to classroom. And then if you really don't know something, maybe take a look at it really quickly and then put it away, take a deep breath, and take the exam. You're going to do a lot better mentally for yourself if you do that. But yeah. Are there any? Sorry, go on.
I was just gonna ask if the lab and lecture portions are on the same day
Yes.
of the exam.
Yes. So you'll have your 3 hours in class, 3 hours and 10 minutes technically. You'll walk in, you can choose to do the lecture portion first or the lab portion. Once you take either or, you'll turn it into Dr. B and she'll hand you the other one. So like if I walk in and I decide to do lab first, just to get it out of the way, I'm not stressed about it. If I walk in and do lab, I'm gonna do the lab section completely, and then I'm going to hand it in. And then she's going to hand me the lecture portion of the exam, and I'll sit down on the other side of the room and fill that out and then give it back to her. I don't know if you guys are like this, but I did the lab personally first because I found that it was a little bit more exhausting. And if I were to be tired doing the lecture portion of the exam, I had a 25% chance to get the right answer. So I would have rather spent more time and more energy on the lab portion to get those points, because there was no making up those answers. There's no like guessing on those. It was either you know it or you don't. So I would rather have spent more time on that and then guessed on another question that I had, not even the slightest clue about or something.
Mhm.
That's what I personally do. If you have the stamina for those types of tests, go for it. Do whatever first. But yeah. So how do we feel? Do I have any other questions?
I feel pretty good. I don't think I have any other questions.
Okay, good. Just so you guys know, Zoom likes to kick me out at 40 minutes exactly, almost every single time. So Zoom just said you have less than a minute. So if it kicks me out, then if you have any other questions, you can send them to me on Remind. You can shoot me an email. I will try to get back to you guys as soon as possible. But if you're Tuesday, Thursday, I'll see you in class, okay? You guys are free to go. But yeah,
Great. Thank you so much.
yeah, no problem.
Have a
Bye.
good day.
Have a good
Bye.
one.